Good morning. Today is Wednesday, May 6th, and we are to our second to last lecture. So today we are going to continue our discussion of the reproductive system. We just have a little bit left to finish off for the male, and then we get to dive into the female reproductive system. So again, we're wetting our feet in the kiddie pool, and then we'll dive into the deep end. As I talked about in the last class, I think it is going to be good. I think it was helpful last time, so there was a lot of positive feedback on it. So I think it is worthwhile to do another histology review. I know I go through some of the histology as we're going through the lecture, but having that extra time with it, I think, can be useful. So we will be doing that review on Friday from 10 to 11. I think that was the time we did it last time as well, so I think that worked well. We'll stick with that. You also have only one more assignment due, your Unit 27 review, which is due on Monday, our last lecture. The format has been set for all of the exams. Your lab and lecture exam is going to be 1 week from today, and that is going to be available from 7 a.m. to 2 p.m. The format for that is exactly the same as the previous one. Overwhelmingly, the people that responded to the review, to the lab preference, preferred the presentation of all of them at once. So it'll be the exact same format of what you guys had for the digestive system. So that is going to be the format for that. And again, you can take them in either order you want. They are available from 7 a.m. to 2 p.m. and both exams must be completed during that time. Then you get a whole week to study and prepare for your final exam. The final exam is going to be 100 multiple choice questions, and you will have 100 minutes to complete it. I am not going to be as cruel as to force you to take it within the 2-hour finals window that we would normally have available to us, but I am going to limit it to our class time to make it a little bit more restrictive that way. So you must complete it between 7 a.m. and 11:35 a.m., which is our normal class time for this. So again, it'll take a little over an hour and a half. 100 multiple choice questions must be completed during that time. So make sure you start with enough time to be able to complete that. And then hopefully we'll have the grades finalized shortly after that. All right, any questions on any of that? Like I said, we're running out of things to talk about because we are rapidly approaching the end of this class. All right, stunned silence, exactly what I love first thing at 8 o'clock in the morning. So let's go ahead and switch from this then and talk about our anatomy and physiology. Switch to the lecture slides and get rid of my scruffy face. All righty. We left off last time and we were working through the, where's my annotation token? There it is. We were working through the male reproductive system. We had talked about the testes. We had talked about the ductwork system, most of it. We had talked about both the production of sperm as well as the accessory components to it. And so, as I said, really all that really leaves us is the external genitalia for the males as far as the anatomy goes. And of course, what we're left with is the penis. The penis's job is dual function in that it plays a role in both the urinary system and in the reproductive system. In the urinary system, it allows you to write your name in the snow. And of course, in the reproductive system, its job is to be the copulatory organ. Its job is to present the ejaculate, the semen, to the vaginal canal to be able to hopefully fertilize that egg and produce offspring. The penis has several regions to it. The prominent shaft or body of the penis. It has an enlarged head known as the glans region. And while this illustration does not show it, the next one we're going to look at does, there is an extension of skin that covers the glands known as the prepuce, or what is commonly referred to as the foreskin. That foreskin is typically removed for health, for cultural, for religious reasons in a process that is called the circumcision. Those are the external components of it, but the functional comparts are truly internal, and that's what we see here highlighted on the inside. What allows the penis to be a copulatory organ is that there are three distinct regions of erectile tissue that turn it from the flaccid structure that we see in this illustration to the rigid copulatory organ that we need it to be. Here is that other picture I was talking about. This is kind of a longitudinal section through the penis. Notice here we actually see the prepuce is left intact where it is covering the glands of the penis. But what we can also see nicely from this now are those three erectile tissue regions. If instead of a longitudinal section, we take a cross section, we get a better sense of the organization of these areas of erectile tissue. The first, illustrated here in red, is what is known as the corpus spongiosum. The corpus spongiosum is what surrounds the urethra, or really, more specifically, what we should say, it surrounds the spongy or the penile urethra. Oops. A portion of the urethra, the part that goes through the penis. Its primary job is to maintain the opening of that urethra. Again, the job of the penis is to be the copulatory organ that presents the ejaculate to the female's vaginal canal. Intercourse, when done properly, should be vigorous, and as such, we do not want that urethra to collapse, because if you squeeze it closed, then a release of the ejaculate is not possible. So by having this rigid structure that surrounds the urethra, it helps to maintain the opening of the urethra so that ejaculation can take place. Notice, though, of this region, again, it is that hollow tube that we see, the urethra there, that is surrounded by that narrow corpus spongiosum like we saw in the cross section. But if you notice distally, that is what enlarges. The corpus spongiosum enlarges to form that enlarged head or glans region. And notice also there is a proximal enlargement of it as well, known as the bulb. And if you notice, that bulb happens to be where the bulbourethral glands, ducts, enter into the penis and release as well, which is why they're called the bulbourethral glands. That's convenient and it makes sense. The other two structures, these two that are larger, they are superior and they are lateral to the corpus spongiosum, are what are known as the corpora cavernosa. Of course, corpus is singular, corpora is plural, cavernosum is singular, cavernosa is plural. So again, fun with vocabulary. But these two large structures, which are sometimes referred to as the dorsal bodies, but that is not a term you're allowed to use on the exam. You will use the appropriate anatomical term, corpora cavernosum or corpus cavernosum for one of them, are what give the penis its rigidity and again allow it to have that penetrating force as that rigid copulatory organ. Oops. Notice also, and we'll talk about the physiology of this briefly, there are deep arteries located within this, and those are what release the blood into this erectile tissue to cause the engorgement. And we'll talk about the physiology of that in just a minute. There are specializations to the corpora cavernosa as well. Notice at the proximal end, they taper down into these tail-like structures known as the crura, crura, of course, being plural, crus being the singular. And if you notice the two crura and the bulb, these three specialized structures of erectile tissue collectively are what form the root of the penis. And the root of the penis is what basically anchors it to the pubic arch and to the urogenital diaphragm. So basically, these are the structures that anchor the penis, attach it to the outer wall of the female body. All right, now that we've talked about the structures, let's talk about the function. Oh, wait, hold on. I see there is an answer, a question. On the anatomist, it says central artery. Is that the same as the deep artery? Yes, the central artery and the deep artery are the same. And as you can see from the picture, you can see why they're referred to as both. It is at the center of the erectile tissue. It is deep inside the erectile tissue. So central artery, deep artery, those are both appropriate terms. Thank you for asking that. All righty. I know first thing you want to talk about 8 o'clock in the morning is arousal and ejaculation. A good mnemonic to help you to remember this, a fun mnemonic that you should take home to your family and friends, is point and shoot. Right? Point stands for the arousal, and it is a parasympathetic reflex. Shoot stands for sympathetic, and that is the ejaculation, which is a sympathetic reflex. So as we talked about a little bit before, and we'll continue to talk about if you think back to 431, we were talking about the autonomic nervous system. One of the things we talked about is arousal, or tumescence, the formation of the erection of the penis, is a parasympathetic reflex. Right? We talked about this in great detail in 431. Right? You, again, back in ancient times, used to be able to date. And so on that all-important third date, you would take your man out to a very fancy restaurant and buy him a really nice meal. Of course, you expect something in return for that really nice meal, and he knows that. So he may be a little bit nervous, and as you know, if he's a little too nervous, that can affect performance, and that's because arousal is a parasympathetic reflex. As I'm sure you recall from 430, most of the blood vessels of the body are controlled solely by the sympathetic nervous system. The one exception are these blood vessels located in the erectile tissue. That means the penis in the male. It also means the clitoris and the labia in the female. And we'll talk about that anatomy and arousal on, I guess, well, I don't think we're going to get to it today. So my guess is we'll do that on Monday. But upon arousal, what happens is our parasympathetic nervous system causes the release of nitric oxide. As I'm sure you recall, nitric oxide is a vasodilator, and it causes that deep artery of the erectile tissue to enlarge. It also causes the capillaries in that region to become extra leaky as well. And so what happens is blood flows to that area, leaks out of that area, and fills that erectile tissue, which is essentially acts like a sponge. If you think of when you go to do the dishes and you've got that dried up kitchen sponge sitting on the countertop and you take it and you put it in water, it absorbs that water and it enlarges as a result of that. However, it gets a lot bigger, but it's also very floppy as well. And again, that wouldn't be very useful as a copulatory organ. So yes, the tissue does engorge with blood and it swells as a result of that. However, and we didn't point it out on the previous picture, but we can sneak back and look at it, and then also we would look at it when we talk about the histology as well. There is a collagenous fibrous, basically mesh on the outer surface surrounding the erectile tissue called the Tunica Albuginea. I know that sounds familiar because it is. I don't know why that's all in capitals. And this Tunica albuginea is this collagen-rich fibrous structure that basically puts a mesh around the outer erectile tissue, so that as the erectile tissue swells, it pushes against this collagenous border, and that is what causes it to become turgid, causes it to become rigid, and allow it to be the copulatory organ that we need it to be. The other advantage of this swelling of the erectile tissue, and in particular the corpora cavernosa, is as it swells, that swelling of the tissue compresses the drainage veins. So the tissue becomes congested with blood. It fills with blood, and that blood cannot be released. Right? And that, again, congests the area with blood. It pushes against that rigid collagen structure, and you get the turgid copulatory organ that the penis can become. Now, again, I know this is one of those things we always talk about, how as we learn how anatomy works, we use these things to our advantage. And there is I hate to use the word toy, a sex instrument that is sometimes used that takes advantage of this, where a ring will be placed at the base of the penis when it is flaccid, so that as it engorges, it helps to increase the compression of those drainage veins and can maintain the rigidity of the penis for a longer period of time. those things, while they sound fun on paper, are incredibly dangerous, because the problem is that if it is not, and these things typically aren't one size fits all, if it is too restrictive, then after copulation, drainage still cannot occur. And there have been numerous cases of individuals who've had to gone into the emergency room, and what they have to do in those situations is take a large bore needle, insert it into the erectile tissue of the penis so they can extract some of the blood to decrease the rigidity of it, to be able to remove those sex toys, right? And as we've talked about, this tissue does not regenerate. It heals via fibrosis, which means it is going to scar. Erectile tissue is very specialized tissue, and if you damage that erectile tissue extracting the fluid, then that can affect the ability to produce an erection on future attempts. So again, it is not something that someone should consider doing. But on its own, it is capable of maintaining that rigidity to allow copulation to occur. Remember also, when we talk about arousal, it is not just the engorgement of the erectile tissue. That's really two things, and we'll see something similar in females as well. Basically, arousal has two components. Really three, actually, let's lie. Three components to arousal. The first is engorgement of the erectile tissue. The second is glandular secretions. And again, in the case of the male, that is primarily those bulbourethral glands. Remember, we talked about how they produce a thick, clear, viscous, alkaline secretion, mucus-rich secretion that again acts as a lubricant, acts as a neutralizer for the acidity of the urethra, as well as the acidity of the vaginal canal. But the other thing that occurs as well are smooth muscle contractions. And again, I make a point of emphasizing these three factors because we're going to see the exact same three factors occur in females as well. That contraction of smooth muscle is seen in the ductus deferens. Remember, we talked about at the ampular end of it, you're going to see an increase in the peristalsis there, not as prominent as when ejaculation occurs, but you can see an increase in the peristaltic contractions that are occurring there, which is why some sperm will be released in that pre-ejaculate. Again, I can't emphasize it enough. Withdrawal is not a viable method of birth control. But you also can get other smooth muscle contractions. The hair can stand up on end upon arousal, so you can see erector pili muscle contractions. The nipples can become erect in both males and females upon arousal, and things along those lines from that smooth muscle contractions as well. So these are things that are seen in males, and then we'll see them in females as well. Clear that, go there. That arousal also then triggers a positive feedback process. Again, way back in 430 when we talked about positive and negative feedback, the two examples everybody seems to always come up with for positive feedback processes are blood clotting and parturition, the giving birth of the labor of a female. But a third prominent positive feedback process in humans is ejaculation. This is a sympathetic reflex where basically you get stimulation and arousal continues to enhance these sensations. That increases the sympathetic input to the reproductive region, which increases the excitement and enhancement of it, which increases the neural input to it, and on and on and on until that critical point is reached and ejaculation occurs. And once again, with similar to arousal, we are going to get with ejaculation, we are going to get glandular activity and muscular activity. As a result of this. So from a ejaculation occurs, we get the contraction of the internal urethral sphincter. During this process, the muscular activity, we don't want the semen to go up into the bladder. Instead, we want it going out the urethra. So that is going to constrict. So it's not possible to urinate and ejaculate at the same time. The semen is going to be released from the ampullas of the ductus deferens with powerful contractions that will release a large volume of sperm into the ejaculate. But it is also going to cause the release of the prostate and the seminal vesicle secretions as well. All of these are going to be expulsed into the urethra and then are released with powerful peristaltic contractions that expulse the semen from the penis. And if you've ever seen the release of semen from a penis, you know it's not like turning on a faucet. It is in a pulsatile nature because of that peristalsis that is taking place within there. So glandular activity, muscle activity, smooth muscle in the internal urethral sphincter, smooth muscle within the urethra for that peristalsis. But there is other contractions that are taking place during this time as well that lead to the loss of the erection, or a process we call detumescence. Part of that sympathetic, after the ejaculation, that enhanced sympathetic input causes a constriction of those blood vessels. So remember, we released the nitric oxide, and that caused the dilation of the blood vessels, that caused the engorgement of blood into the area. Well, now we are going to get a constriction of those blood vessels. So now less blood is flowing to the area. Now remember, as we talked about, that area is congested, and that congestion is helping to keep those drainage veins constricted. But there is smooth muscle within the walls of that erectile tissue, and that smooth muscle will contract as well. And that contraction actually helps to force open those drainage veins and expulse the blood from the penis, and it loses its rigidity. This period of detumescence, can vary in how long it lasts. Again, part of it is how long and how strong that sympathetic activation is. But in most cases, it lasts till pretty much the end of Westworld, and then everything is back to normal, or you're asleep, one or the other. All righty. Questions on that? Excellent. Now we can all smoke a cigarette. All right. Now that we've talked about the fun stuff, let's talk about some of the less fun physiology. And that is our estrogen and androgen production, and along with our sperm production as well. As we will see, the male cycle is much less complicated than the female cycle. There is a male cycle indeed. However, the male's hormone cycle is minor in fluctuations and is daily, where there are typically hormone levels are higher at the beginning of the day and slightly lower by the end of the day. But for the most part, and the key to it is that it is consistent from the onset of puberty to, like we said, 15 minutes after you are dead. There may be a slight decrease in levels over time, over long periods of time, but for the most part, it is pretty minor in that. Now, let's talk about some of the things we do know. Of course, the androgens, primarily testosterone, is produced, of course, by those interstitial cells. And again, because I like saying it, of Leydig, but it isn't required. And remind me again what hormone is responsible for regulating androgen production?
Follicle-stimulating hormone.
There you go. Excellent. Follicle-stimulating hormone is the one that stimulates the interstitial cells and causes those interstitial cells to produce androgens. Excellent. Oh, wait, no, hold on. Sorry, I'm sorry. That is not correct. I'm not thinking. No, remember, a follicle-stimulating hormone is the one that causes sperm production. I'm sorry. I was busy writing, wasn't paying attention to what I was saying. Never a good thing. It's actually luteinizing hormone. Remember, the way I remember it is luteinizing hormone causes the production of progesterone and testosterone. So the two rones are produced by luteinizing hormone. That's the way I remember it. So I apologize for that. I wasn't, like I said, I was going with the flow, wasn't thinking. That luteinizing hormone produces androgens. Remember, those androgens do not affect sperm production, right? Not affect sperm production. However, those androgens are needed for sperm maturation in the epididymis. So in the epididymis, androgens are going to help to make the sperm viable. And again, by viable, we mean capable of fertilizing an egg. Androgens are necessary for maintaining our reproductive organs. They play an important role in our puberty, that metabolism, that growth, turning boy-type people into men-type people. And part of that is expressing those secondary sexual characteristics, the production of secondary hairs, buccal, thoracic, axillary, pubic, right? The increase in cartilage, the thickening of the vocal folds, right? All those other things, increase in bone and muscle mass, all of those things we associate with that conversion from boy-type person to man-type person. And it plays a role in sex drive and aggression as well. Let's not forget an aggression. All right, so we got all of that. How it is regulated, and I love this picture. It is pretty simple and straightforward. As I said, the male pattern of secretion is continuous, basically from puberty till death, and there is a minor daily fluctuation in this, but for the most part, it's fairly stable and fairly consistent. This picture does a nice job of doing this, but let's actually go through it together. Let's switch to the whiteboard and do a little drawing. Of course, we know, I don't want this to be red. If we are talking about hormones, we know that, of course, that hormone control begins with the hypothalamus. And again, let's stick with black for identifying things. The hypothalamus, of course, communicates to our pituitary gland. But in this case, if we're talking about controlling the testes and their function, it's going to specifically be the anterior pituitary. And remind me again how the hypothalamus communicates to the anterior pituitary.
Is it hormonally?
Absolutely, it is indeed hormonally. So let's sneak that in there as well. Right? Remember, it's going to release, it's going to produce and release those releasing and inhibiting hormones. So let's say it this way because that's confusing. It uses releasing and inhibiting hormones. But for purposes now, let's go releasing hormones. And more specific, it's our gonadotropin releasing hormones. So let's, because I need some of that space. Trying to think ahead as we do this. Sneak that out of the way. Move this here. And then we can sneak that in there. Excellent. So in particular, the hypothalamus in this particular instance is going to release gonadotropin-releasing hormones to the anterior pituitary. And that is going to cause the anterior pituitary, and let's give ourselves a little bit of space here. It is going to cause the anterior pituitary to release two types of hormones, two gonadotropins, and those are, as we've already talked about, our luteinizing hormone and our follicle-stimulating hormone. Now, as we just finished mentioning, that luteinizing hormone is going to target the interstitial cells of the testis. And so let's put a box around that. And it stimulates these interstitial cells to produce androgens. All right. Now, if you remember, when we talked about androgens, we mentioned that hormone regulations is primarily controlled by what again? What is hormone regulation primarily controlled by? Cumulative class, potential question on the final exam. Almost certain to be a question on the final exam now. Come on.
Chemoreceptors?
Close. What would those chemoreceptors be looking for?
Stuff in the blood.
Yeah, what stuff in the blood? You're absolutely correct. The androgens themselves. All right. Remember, we talked about how hormones are primarily regulated by negative feedback. So what happens is you produce androgens. Those androgens are released into the blood. The amount of androgens in the blood are regulated by the hypothalamus, and that determines how many gonadotropin-releasing hormones are produced. Think of it this way. If we're producing too many androgens and the androgen levels were to go up, so if androgen levels went up, our hypothalamus, let's make that green so it's consistent. If our androgen levels increased, our hypothalamus would see, oh, there's too much androgens in the blood. It would produce less gonadotropin-releasing hormone. And if it produces less gonadotropin-releasing hormone, what happens to the amount of luteinizing hormone that is produced by the anterior pituitary? We get a decrease in luteinizing hormone that is produced and released by the anterior pituitary. And what happens to androgen levels? Less androgens are produced, and androgen levels come back into balance. That makes some semblance of sense?
Yes.
Yeah, and the reverse is true as well. If androgen levels are too low, our hypothalamus is monitoring that in a negative feedback process, sees, oops, there's not enough. So you're absolutely right. The thing it's looking for in the blood is the androgen itself. Notice this is an example of humoral control, right? It's not that the androgens are stimulating the hypothalamus. The hypothalamus is counting how many of those androgens are in the blood. That's that humoral control we were talking about. And that's really what negative feedback is. Negative feedback is counting how much of something is in the blood. And if there's too much, let's make less of it. And if there's not enough, let's make more of it. That's really all it is. It's super simple. That left side of this graph, that's all that's going on. We're monitoring the negative feedback. We're monitoring the amount of how many androgens are present and affecting androgen production based on that. The right side of this is a little bit trickier. Follicle-stimulating hormone stimulates what cells? Oh, wait, hold on. I don't want that to be red, green. Let's go back to black. Follicle-stimulating hormone stimulates what cells?
The nerve cells.
Exactly. Excellent. Perfect. Follicle-stimulating hormone stimulates the nerve cells. Oops, that also should not be green. And it stimulates the nerve cells really to do three things. There are three things that this nerve cell is going to do. The first and most obvious thing it's going to do is it stimulates the spermatogonium. And I guess it should be plural. Stimulates the spermatogonia, and of course, we make sperm. And it should be spermatozoon, but I don't have the space, so I'm just going to cheat and write sperm. All right? So the more follicle-stimulating hormone, the more the nerve cell stimulates the spermatogonia, the more sperm we make. But remember, the nerve cells have other responsibilities. Like any good nurse, it does a lot of stuff. And in this case, one of the other things that it is doing, it is producing fluids that are going to help to maintain the sperm. And remember, those fluids produce the fluid pressures that propel the sperm through the seminiferous tubules. But remember, it also is going to produce those androgen binding proteins so that we can carry some of those androgens into the epididymis so that those sperm can mature. However, the third thing that our nerve cell does is it produces its own hormone, inhibin. And as we talked about, inhibin's job is to, not surprisingly, inhibit things. I'm going to cheat by bringing this straight up this way. Actually, hold on, I want to bring that all the way up. There, perfect. Inhibin is a hormone, and it provides hormonal control of the anterior pituitary. And with a name like inhibin, guess what it does to the anterior pituitary? Anterior pituitary?
Inhibits.
It inhibits it, absolutely. It inhibits the production and release of gonadotropins. It also, and I'll cheat and do this one as a highlighter, can influence the hypothalamus as well. So it primarily influences the anterior pituitary, but it can also inhibit the hypothalamus as well. So again, as we talked about, making sperm is important, but making more sperm faster, as we talked about, isn't necessarily a good thing. So if we are producing, let's say it this way, if there's too much follicle-stimulating hormone, then as a result of that, we will be, and now I have to cheat and erase that so I can rewrite it, producing sperm too quickly. But the other thing that will happen is that if there's too much follicle-stimulating hormone, not only are we producing sperm too quickly, but what happens is we also get an increase in inhibin production. And more inhibin means more inhibition. And we are going to produce and release less follicle-stimulating hormone, and our system is brought back into balance. Notice this on the right side is an example of hormonal control. So those two control processes we learned in the endocrine system, we get to use and understand again. Again, that's it. And it may look intimidating as you look at it, but as you can see, it is a very simple, very straightforward process where the goal isn't to produce as much of these things as possible. We don't want to have massive amounts of androgens. We don't want to have massive amounts of sperm. We want to produce just the right amount of both. And this system is set up to basically stabilize that. So it maintains that consistent control, consistent levels. None of these things should get too high. None of these things should get too low. And if that's the case, we're going to continue to produce androgens at a consistent level. We're going to continue to produce sperm at a consistent level. And that's it. End of story. It really is just this simple for males. We really are just that basic. All right. Now, we've done this here on the whiteboard, which was a tremendous amount of fun, but let's look at the pretty pictures from your textbook, and we can see this exact same thing. Again, we have this continuous pattern from puberty to death. We are constantly producing luteinizing hormone and follicle-stimulating hormone from the pituitary. Notice, as we talked about, where's my annotation? There it is. And that went away too. Perfect. All right. Their release, production and release is, of course, controlled by the hypothalamus, which is releasing that gonadotropin-releasing hormone. So the hypothalamus releases gonadotropin-releasing hormone to the anterior pituitary. The anterior pituitary produces and releases follicle-stimulating hormone and luteinizing hormone. Luteinizing hormone stimulates the interstitial cells to produce testosterone, which is controlled by a negative feedback process. Follicle-stimulating hormone stimulates the nurse cell, or sustentacular cell, or Sertoli cell. Remember, all of those are acceptable names for it. Its job is to produce androgen-binding protein, to cause the maturation of the sperm, that process of spermatogenesis, which they've written right here, and that nurse cell produces inhibin, which inhibits the anterior pituitary as well. Now, the one thing I would say is notice this simple illustration shows this dotted line going back to the anterior pituitary. However, if we look at the picture from your textbook, and we'll come back to this in just a second, but it says all the things we said before. Notice here, we more clearly see that testosterone influences the hypothalamus. Inhibin primarily affects the anterior pituitary, but also can indirectly inhibit the hypothalamus as well. So notice that previous picture shows both going to the anterior pituitary. That's not truly the case. Testosterone levels are monitored more by the hypothalamus than by the pituitary gland. Which you think about makes sense. The hypothalamus is the one that is neural and hormonal. It's that mixed gland, so it's our regulatory. So it makes sense that that's the one that would be monitoring levels of testosterone. And that's what we have, that negative feedback from monitoring androgen levels, whereas the Sertoli cell, this nurse cell, the sustentacular cells, stimulate spermatogenesis, produce antigen-binding protein, and produce and release inhibin. That affects and decreases production and release of both luteinizing hormone, follicle-stimulating hormone, and can indirectly decrease gonadotropin-releasing hormone levels. And the goal of this system with all these feedbacks is to maintain a consistent level that, again, might vary slightly during the course of the day, a little higher earlier in the day, a little lower later in the day, and also may slightly decline with age, but does not have to. And again, here you see the pretty picture from your textbook, which essentially shows exactly what we drew on the whiteboard. All righty, questions on that? All right, excellent. No questions, because again, boys are just that dumb and simple. Excellent. So let's move on to something much more interesting, and that is the female reproductive system. Now, again, obviously these are related systems. It's all reproductive systems, so there is obviously some similarity. We have our primary organ, and the primary organ of the female reproductive system are the gonads. In this case, it is the ovaries. We also have a ductwork system, tubes connected to this for a passageway for these gametes that are produced by the ovaries and by the males, the sperm. They include the uterine tube, the uterus, and the vagina. However, and it's not truly emphasized on this picture, but one of the points that I want to make sure to emphasize, and we'll emphasize again and again, is this is an open duct system. It was completely closed in the male. That sperm, once it was produced in the lumen of the seminiferous tubule, was connected duct by duct by duct, right? Straight tubule, rete testis, efferent ductule, epididymis, and on and on and on and on. There was one and only one place it could go. What we see here in the illustration are these finger-like extensions of the uterine tube, known as the fimbria. These move in a wave-like fashion over the top of the ovary, but don't actually connect to the ovary. So while it is a hand placed on top of it, it does not actually connect to it. So it does things, those wave-like motions that are produced by the fimbria produce currents in the interstitial fluid to help to draw the egg into the uterine tube, but it's not required to go there. There's nothing stopping the egg from floating around in the abdominal pelvic cavity. Of course, if it floats around in the abdominal pelvic cavity and doesn't go in the uterine tube, that dramatically decreases the likelihood of being fertilized and being brought to term, but that's beside the point. It doesn't have to go in there. By the same token, and that's what we keep talking about, pathogens, sexually transmitted diseases that get into the female reproductive system are not limited to the female reproductive system. They also have the ability to get into the abdominal pelvic cavity. And so typically sexually transmitted diseases are a much more serious concern for females than they are for males. And then, of course, we have the external genitalia of the female as well. So again, those three main parts, just like we have the three main parts in males as well. Like with males, we will start first with the primary organ, and those are, of course, the ovaries. Their functions are, like the testes, twofold. Their job is to, their job is to produce the gametes and produce hormones. Unlike the testes, though, remember, it is capable at different stages of its cycle that it can produce two classes of hormones. Whereas, remember, our testis only produces androgens. Although, really, you could argue three. Put three in parentheses. Oops, it's not parentheses. Classes of hormones. Again, incredibly impressive in their function, but once again, not super impressive in their appearance. They are lateral to the uterus. And again, internal, inside of the female body, similar in size to the testis, about 1 inch by 2 inches in size, only weighs about ½ an ounce or something crazy like that. One of the things that we've talked about, one of the big, huge sexual differences between males and females is that the females house and provide for and sustain that developing baby for those 40 weeks of development. And so as such, the internal structures of the abdominal pelvic cavity and the reproductive structures have to be a lot more fluid in their, or flexible. Flexible is a better word. Flexible in their anatomy so that they can expand to accommodate that basketball inside of them. We see this in particular with the location of the ovaries. They're kind of generally held in place by this extension of the peritoneum. The peritoneal cavity kind of forms up these connective tissue structures that are collectively known as the broad ligament that help to hold the ovaries and the uterus in place. They are suspended laterally, anchored to, again, loosely anchored to the lateral walls of the abdominal pelvic cavity by the suspensory ligaments. There is a Mezzo Varium, which suspends the ovary between the ovary and the uterine tube. And there is a mesometrium that is between the ovary and the uterus. And these two structures, the Mezzo Varium and the mesometrium, are together part of, they're not the entire, there are a couple other structures now responsible for, but they are the two components that you're responsible for that help to form collectively what is known as that broad ligament. All righty. Lastly, the ovaries are also loosely anchored to the uterus by a structure called the ovarian ligament as well. So all of these are ligaments that help to support. Here we see them in name. I think in many ways it's easier if we see the relationship to this. Normally I'm drawing them on the boards while we do this, but it wouldn't defeat the purpose. When I do it, it looks like a big moose anyway. So let's actually take a look at the, oh, and of course the round ligament, your fifth ligament you're responsible for is what anchors the uterus. But I think where to see this easiest is when we look at the illustration from your textbook. So again, here is that medially located uterus, and lateral to them, here we see the ovaries. As we mentioned, these need to be loosely held in place, but they need to be flexible in their ability to move, because this structure has to expand to be able to accommodate a basketball. So we see here the ovarian ligament is the ligament that helps to anchor the ovary medially by attaching to the uterus. Laterally, and here we see the tip of it here, we have the suspensory ligament that anchors it to the lateral wall of the abdominal pelvic cavity. And then here, as I mentioned, we see the two structures that collectively, with some others you're not responsible for, form what we call the broad ligament. They are the Mezzo Varium. Notice the Mezzo Varium is the extension of the peritoneum that basically supports the ovary to and connects it to the uterine tube. And the mesometrium, which basically is this inferior extension of the peritoneum that comes up and supports the ovary inferiorly as well. So we have a superior, inferior, medial, and lateral support structure. Notice also this is a posterior view. So if you notice from the starting at the uterus and moving forward, we also see the round ligament as well. The round ligament does not support the ovary. It instead helps to loosely support the uterus in place. If we cheat and go back, I think the next picture has it as well, but I saw it on the previous one, so I wanted to emphasize that here. Notice here we see that round ligament coming off of the front of the uterus to stabilize it in place. Here's that suspensory ligament. Notice the broad ligament is missing, is lacking from this illustration here, but we do see those other two ligaments that we wanted to identify. Ah, okay, it is the same picture. I wasn't sure. Notice what happens then, because of this. Basically what happens, as I mentioned, is the peritoneum extends up from the inferior portion of the abdominal pelvic cavity, wraps around the uterus, wraps around the ovaries, forming this dividing structure that divides the abdominal pelvic cavity on the inferior side. And notice there are two basically invaginations, two sides, the front and the back, pouch-like structures that are formed by that broad ligament and these other suspensory structures associated with the uterus, the uterine tube, and the ovary. And of course, because anatomists love to name everything, they've given these invaginations name. The pouch-like structure that is located between the uterus and the bladder is known as the vesicouterine pouch, and the invagination located between the rectum and the uterus is the rectouterine pouch. So we have these two divisions down deep inferior in the abdominal pelvic cavity that is formed by all of these support structures. And again, notice these are all ligaments, these are all membranes, because, like I said, this needs to be able to expand, and these things need to be flexible in their movement as you grow a basketball inside of your belly. Here we see a pretty picture of that ovary. Like I said, it's about 2 inches by 1 inch in width, 2 inches in length. Like I said, it is tiny, not impressive to look at. What we can see here now, this is not a human ovary we are looking at here, but what is really cool about this particular picture is that this is, my guess is probably cat, is what we are looking at here. And what they often do with these is they stimulate this with hormones ahead of time, so we get massive development and growth of the follicles. We talked about that a little bit when we talked about the endocrine system. And remember, when we talked about the endocrine system, we said how the ovary has that candy-coated shell and that chewy nougat center. It has a cortex and a medulla. Notice here it's a little trickier because the follicles are so developed. It sort of looks like there is an actual mechanical boundary between the two, but there isn't. It's just that this one has been so matured. We can clearly see the cortex, which is the area that has the follicles in it, and the medulla, which is the area that does not. However, I will point out, and again, we'll come back to this much later as well. Notice with these follicles, there is one thing that doesn't look like the others. While these look all like big, large balloons or bubbles, this one looks like a bubble that has popped. And that's exactly what's happened. This follicle has ovulated, expulsed its egg. And remember, this structure is in the process of becoming our corpus luteum. Remember, we talked about the ovary and its follicle cells. After the egg is expulsed, those follicular cells are converted into the corpus luteum, which is that important warning bell system to the female's body that ovulation has occurred and pregnancy could begin any minute now. As we can see, there's a lot of stuff going on inside of the ovary. Some of the anatomy is similar to what we see in the male, starting with the fact that we have two external layers to it. We have the outermost layer, which is essentially the visceral peritoneum, and that visceral peritoneum on the outer surface of it is what is known as the geminal epithelium. And much like we saw with the testis, there is the fibrous bulk that forms the structure. That fibrous bulk is known as the Tunica Albuginea. However, remember in the testis, that Tunica Albuginea forms deep septums that divide the testis into lobules. There is no lobules in the ovaries. We just have the two regions, the outer cortex, which as we can see here, again, contain the follicles, and then the medulla at the center, which contain the large blood vessels and nerves. Oops, oh, there you go. And here we see a pretty picture showing that same thing as well. This one happens to be actually, by the looks of it, it's probably rabbit. Excellent. Like I said, two regions, distinctly different in function, but again, there's no anatomical border to them like we've seen in some of the other organs. So we have our medulla, which again is the inner region, contains the large blood vessels and the large nerves. And we have our cortex, which is the outer region, which contains the ovarian follicles And here we see the ovarian follicles at different stages of growth and development. The job of those follicles is to form is probably not even the right word here. I don't like the word form, so let's go ahead and put a big line through that. Instead, really, their job is to mature the gametes. And so let's actually change that. Ah, too far back. There you go. So as I said, these ovarian follicles are where we mature the female gametes and then expulse them so that, again, in the hopes that fertilization will occur, propagating the species and all of that. These ovarian follicles contain the maturing, notice I did not say mature, I said maturing developing gamete of the female, what is known as an oocyte. And in fact, when it is expulsed, it is still immature. So the oocyte never fully matures while it is inside the ovary. Right? If you think about it, it's kind of similar to the testis. The spermatozoa do not mature in the testis. Instead, they mature in the epididymis. Well, it's the same thing here. Our gamete is releasing an immature egg. But the follicle is more than just that maturing oocyte. They are the support cells around them. And these support cells are basically given names, part of them based on their location, part of them based on their function. And so there's basically three types of cells, and we'll identify all of them as we go through them. First are the follicle cells, then they're the granulosa cells, and then there are also thecal cells as well. And so we'll identify all of those, look at those, and talk about the function of these as we go through our ovarian cycle. However, before we can get to the ovarian cycle, we need to talk about the process of oogenesis. Oogenesis is the process of gamete formation in a female. Now, of course, being the sophisticated students that you guys are, of course, you know that when we talk about two closely related processes like spermatogenesis and oogenesis, if we have two similar processes, how many possible essay questions are on that?
Three.
Three, there you go. Describe spermatogenesis, describe oogenesis, and compare spermatogenesis and oogenesis. These are obviously going to be similar processes, but they are very, very different in many ways as well. The good news is that both involve mitosis and meiosis. And so those processes we talked about in male, reminding ourselves of what mitosis was, and going through the process of meiosis, both 1 and 2. That is something we don't have to do a second time. However, how the process of oogenesis works around meiosis and mitosis 2, mitosis and meiosis 1 and 2 is going to be slightly different. So like I said, this is going to be a little bit more complicated process in females than it was in males. But at its core, oogenesis involves the growth and development of the oocyte and its follicle and the cells that wrap around it as well. All right. Like I said, this is a little bit more complicated of a process. It's a teeny bit early, but this is a good natural stopping point. So let's go ahead and take our first break here. And after this break, we will come back and we will describe the process of oogenesis and begin our merry mystical journey around the female's physiology. My watch says 9:04, so let's come back and restart at 9:15. And I will remind myself to start the recording. All right, excellent. Let's go ahead. So any questions before we take our break? All right, out. All righty, we are back. So let's go ahead and get started. So our goal is to discuss the process of oogenesis. And again, along the way, one of the things that we want to do is make sure we emphasize how it is different from spermatogenesis. One of the big ways it is different is when it begins. Oogenesis begins in female, actually in utero, prior to being born, right? Boys being lazy slackers, wait till puberty before they start producing their gametes. Of course, then they make up for it, 500 million a day, but females, being the overachievers that they are, right, while mom is developing the daughter is already working on the grandkids. So the process of oogenesis actually begins prior to birth. Prior to birth, primarily, is when we have the process of mitosis going on. Whoa, what's going on? Mitosis is primarily occurring prior to birth. From there, at the time of birth, it then pauses. Again, pausing isn't something that occurs in the male system. They may take years before they start, but once they start, like I said, sperm production continues till 15 minutes after they're dead. Females pause the process at the time of birth and then start again at puberty. And do females continue to mature gametes all the way up to 15 minutes after they're dead?
Nope.
No, a female process ends and she becomes completely worthless from a reproductive standpoint when our ovarian cycle stops and the maturation of the gametes stop at a process that is called what?
Menopause.
Menopause. There you go. Like how it's the boy who's answering the questions for that. Excellent. The process ends at menopause. And again, the female is no longer reproductively viable at that point. All righty. Now, like I said, let's finish this. Spermatogenesis, remember, has three stages: mitosis, meiosis, and spermiogenesis. We have to make many cells. We have to turn those cells into gametes, and then we have to make them special shaped cells. Oogenesis only has 2 steps. But the first 2 steps are the same: mitosis, and the goal of mitosis is to make many gametes, or many cells, let's say that way. And then meiosis, which is to convert those cells into gametes. But the female gamete, the ovum, is a cell-shaped cell. So there's no specialization of that cell that has to occur at the end of that process. That doesn't mean there isn't special stuff happening along the way. There's a lot of special stuff that happens along the way. And we'll see that as we work our way through the process. Now, the other point I want to emphasize, and again, we'll see this in the hinting at this with this picture here, because notice we're only getting half of the chart. So the other thing to remember that is going on as well is that at the same time that we are getting changes that are occurring within the oocyte, there's also those support cells as well, that follicle that is maturing and growing as well. We will focus on what the follicle is doing in the ovarian cycle. And for right now, we are going to focus just on what is happening with the egg in that process of oogenesis. All right. So like I said, prior to birth, this process begins. And in a similar fashion to what we saw in males, we have a stem cell. I'll cheat and put it over here. And in this case, the stem cell is called an oogonium. And that oogonium undergoes mitosis. And of course, as we know, what that means is one cell becomes two cells. And those two cells are identical to the original and to each other. Exactly what we saw in the male process, right? And again, as we see, we're going to have type A and type B. Type A becomes stem cells again. And type B are going to start the process. Well, let's say it this way. Become primary oocytes. Wow, there's a lot of typing errors in there, but you get what I'm saying. So this process continues, dividing, producing new stem cells, producing new primary oocytes, and this process continues until birth. At birth, there are no longer any stem cells. The only cells that remain are primary oocytes. So notice once a female is born, she is born with all of the gametes she will ever have. And if you look, that number is a surprisingly unimpressive 2 million. Now, I know 2 million of anything doesn't necessarily sound unimpressive, but when you think of the scale between what a male makes and what a female makes, heck, I've probably made more than 2 million sperm since the beginning of lecture. Heck, I probably did that during break, right? However, again, while that does not seem like a large number, remember with the goal of all of this, as we've talked about, the reason so many gametes are produced is genetic diversity. Remember, we talked about how when you make these gametes, the way you line up the chromosomes gives you over 8 million different possibilities of how you combined your genetic material. And that doesn't even include the crossover, which gives you a near infinite number of combinations in producing your gamete. And then when you put those together with the near infinite possible variations of your significant other's production of gametes, each time you produce an offspring, they are unique in their own unique way. 2 million doesn't sound like a lot, but let's think at the same time how many a female actually needs. Again, we're going to paint with broad strokes here, but a female cycle is about a month. And how many oocytes are released during that month?
One.
One. So one a month, right? So obviously that's 12 a year. How many years is a female sexually viable for?
About 40.
I like 40. 40 is a good, easy number, right? Again, it may even be a little high, but 40 is a good number. Let's say 13 to 53. That sounds reasonable. So that's 40 years. So 12 a year times 40 years. How many gametes is she releasing? And again, that doesn't include pregnancies. It doesn't include everything. We're broad stroking it here. But if you look at the math of that, that's less than 500. Right? 4,800? My math right on that? So 2 million is more than enough genetic diversity. Even though that number seems unimpressively small, it is still far more than she is ever going to need or use during the course of her life. So still gives that massive genetic diversity. So it is still an incredibly impressive number when we look at it at that point. All right. Let's go ahead and erase that one. Perfect. All righty. So, oh, and I guess I got to erase the rest of this. Go ahead and clear that. So, at the time of birth, there are no female stem cells, no oogonium that are present after birth. All that are left are those primary oocytes. Now, those primary oocytes begin the process of meiosis 1. After all, we know it goes straight from mitosis into meiosis when we were talking about spermatogenesis. And the same things happens here. Every single time we produce one of these primary oocytes, they begin, notice the key word there, begin the process of meiosis 1, but do not complete it. Instead, they actually freeze during prophase, and again, let's emphasize this, during prophase 1. So basically, these cells get to prophase 1. And if you think about when that is, our chromatin is condensing down into chromosomes, which is smart because then that protects the DNA. And maybe the nuclear envelope starts to break down a little bit. Our centrioles are starting. But again, at the very beginning of prophase, prophase 1, the cell starts that process and then freezes. So at birth, a female has about 2 million primary oocytes frozen in prophase 1. This process freezes at birth and then doesn't start up again until we reach puberty. And when's that going to be? What age?
Somewhere around like 12.
Yeah, 12, 13. Let's go with 12.
13.
12 is a fun number. 12, 13. I have 12-year-olds. It's depressing. He's about to be 13, but he's been a teenager for about 5 years, so it's been very, very fun. Now, obviously, 12 years is going to pass by. During that period of time, there is going to be some attrition of some of those primary oocytes. Not all of them are going to survive 12 years till puberty. So by the time puberty is reached and we start maturing these, only about 400,000 remain. So over half of them are lost just during the part of maturing to puberty. But again, when we think of the fact that a female over the course of her life is only going to use about 500 of those, or at least release 500 of those via ovulation at best, it's still a massively large number. All right, so we reach puberty. And when we reach puberty, clear. The hormone process is a little more complicated than this, but we'll start with the basics. As we know, it is follicle-stimulating hormone that stimulates the primary oocytes to begin development. Not all at once, but a handful are going to start the process of developing. And our primary oocyte is going to begin to enlarge, begin to mature, and it is going to typically get to a point where only one of the handful that start the process get to complete meiosis 1. And of course, after meiosis 1, as we know, and I'll sneak this into the bottom, we have 2 haploid cells with replicated DNA, right? So they are NR, as we have talked about. Now, remember, in males, that division occurs equally. They slice the cells in half, and boom, we have 2 cells. Here, as you notice from the illustration, in females, we get an uneven division, where basically both cells get half of the DNA, they're haploid. Notice they went from 2N to N, and again, we can cheat and remind ourselves by putting that R in there to remind us that these are replicated. But here we get this uneven division. One cell gets only the DNA. It gets half the genetic material, but it doesn't get any of the cytoplasm. Obviously, if you don't have Golgi apparatus, if you don't have rough endoplasmic reticulum, if you don't have mitochondria, you're not going to be a viable cell. Instead, this one that only gets DNA, we call the first polar body. The other cell, and let me sneak this in down here. The second cell gets gets half the DNA and all the cytoplasm. It gets all the Golgi apparatus, all the endoplasmic reticulum, all the mitochondria, right? Again, if you think about it, it's not as important with the sperm who gets the Golgi apparatus or how much of it it gets, because remember, eventually that's going to be shed anyway. The sperm doesn't use it. But if this cell is going to become that one fertilized cell that becomes the zygote, becomes the baby, it needs to be able to divide 1 trillion times. And to be able to do that, it needs Golgi apparatus, it needs mitochondria, it needs all of those components. And so this second cell that gets all the cytoplasm as well as half of the genetic material is the secondary oocyte. So again, if you think of what happens after meiosis 1 in spermatogenesis, one primary spermatocyte becomes 2 secondary spermatocytes. Here, one primary oocyte after meiosis 1 becomes 1 secondary oocyte and 1 polar body, the first polar body. And our illustration shows that nicely. And I've snuck it in here on the bottom, but I believe the next page says this as well. All right, so again, the cell divides unevenly, producing that secondary oocyte and the first polar body. All right, so we have this uneven division. However, as it hints here, there is a second big difference between males as well. Those secondary spermatocytes begin meiosis 2 and complete meiosis 2. But that is not the case here. Our secondary oocyte begins meiosis 2. Now, guess what? Not only does our secondary oocyte, but the first polar body can also begin meiosis 2. Of course, let's ask the question, do we care if the first polar body completes meiosis 2? No, because it's basically just excess DNA anyway, but technically it can complete meiosis 2, and we see that here with the picture where our first polar body divides to produce 2 polar bodies as a result of it. But again, just like before, it starts the process, then freezes. It does not complete meiosis 2. It gets a little further this time. Now it freezes in metaphase 2. And it is actually as a secondary oocyte frozen in metaphase 2 that the egg is ovulated. Remember, I mentioned it does not complete the maturation process in the ovary. Instead, it is as this secondary oocyte frozen in metaphase 2 when ovulation occurs. And it mentions here that there are 2 major differences one, the uneven division, one, the fact that it freezes it again. But there's another key, so you could almost think third major difference in this division process. And the third major difference is that the oocyte is not guaranteed to complete meiosis II. The oocyte only can complete meiosis II if it is fertilized by the sperm. Notice here on our illustration, the sperm has penetrated the surface of the secondary oocyte. It now completes its meiosis II. So if that egg is not fertilized, it is viable for about 24 hours. If it is not fertilized during that time, then it will break down and be reabsorbed by the body and never actually completes meiosis II. The only way it completes meiosis II is if it is fertilized by a sperm. So if you notice, this is a dependent process. Right? Like most processes, it cannot be completed without a man. Okay, maybe not. But you get the idea. It is a dependent process. It is reliant on that sperm. Without the sperm, it does not complete meiosis II. Now, notice one last thing. Once again, like we said in number 1, if it completes, if it is fertilized by the sperm and completes meiosis II, again, it divides unevenly, where one cell gets only DNA, and this becomes our second polar body. Actually, I got to move all this stuff up, huh? I don't have enough room. So sneak that up there, sneak that up there, sneak that up there, sneak that up there, sneak that up there. Whereas the second cell, the one with the sperm, becomes the mature ovum. So notice, same process as the male at its core. Mitosis takes us from stem cells to primary cells. Meiosis 1 takes us from primary cell to secondary cell. Meiosis 2 takes us from secondary cell to mature gametes. 1 cell to 2 cells, 2 cells to 4 cells. However, notice in the male process, all the divisions are even, and at the end of that, we have 4 early spermatids that will undergo spermiogenesis and become 4 viable gametes, 4 viable sperm. Here in females, we get 2 divisions, we get 4 cells, but notice only 1 viable gamete. The first division only gives us 1 viable secondary cell. The second division only gives us 1 viable mature cell. So we're still getting 4 cells at the end of this process, but only 1 is a viable gamete. And then obviously the other big difference is this process is continuous in males, whereas here, completion of meiosis II is dependent on fertilization. If that secondary oocyte is not fertilized, meiosis II is never completed. All right, so again, let's go through the pretty words. Again, our secondary oocyte arrested in metaphase 2. It's ovulated, non-functional in that immature state, and it only completes its division if it is fertilized. And when it does, it forms an ovum and a secondary polar body. Notice also where you can have a spermatozoa, a mature male gamete, right, and have it there by itself. You can never have a mature ovum by itself. The only way you get a mature ovum is if it's been fertilized. And if it's been fertilized, is it going to stay an ovum for very long? No. This nucleus is going to pop off. This nucleus is going to pop off. They're going to come together and become a zygote. So it's impossible to just be hanging out with an ovum by itself. When that ovum forms, it within 24 hours is going to become a, less than 24 hours really, within a few hours is going to become a zygote. So again, here we have the pretty picture. If we think about all the different ways that these are different, and there are many, obviously the timing. How long did we say from stem cell to viable gamete? Was it in the male process?
90 days.
90 days, right? And that seemed like a whole hell of a lot at the time, but how long is it in females? At best, and I'm not sure best would be the term, what is the potentially shortest amount of time that you can go from stem cell to a mature gamete?
About 13 years.
There you go, versus that 12 or 13 years, right? And as a father of a 12-year-old, we don't want that necessarily to be successful at 12, right? So hopefully that's not successful till she's at least completed college and has a job, right? House, you know, all those type of fun things. So the timing of that is dramatically different. Obviously, in the males, it is a continuous process. In the females, it isn't, right? The sheer numbers of gametes that are produced, right? The timing, there's a couple ways the timing. When they both start, when they both end. The males, again, is a continuous process where females start early in utero, pause, start again, and then end. There are lots of ways. The number of gametes, from the process, not just total numbers of gametes that are produced over the life or a day or a year, but the number of gametes produced from meiosis, whereas one single meiosis gives us 4 sperm and only 1 egg, and so on and so forth. So there's lots of different ways that we can compare the processes of oogenesis and spermatogenesis and see the difference. Right, and like I said, completion in female, it's dependent versus males, right? Again, remember, the male process doesn't even need androgens. Maturation requires androgens, but just making sperm-shaped cells doesn't require androgens. So pretty much as long as we have that follicle-stimulating hormone, we're going to have sperm. Whereas obviously to get an ovum requires more. All right. So again, 2 related processes, know those 3 possible essay questions. But as I mentioned, not only do we need to talk about what's going on with oogenesis, we also need to know what is going on. We did that already. We need to talk about, while the egg is undergoing all these changes, what's going on its follicle. And it's easiest to tell what's going on in its follicle by looking at the ovary and what's going on in the ovaries. And so when we talk about follicular development in the ovary, right, this, what's going on in the egg, this is our oogenesis. What's happening over here with the follicle, we refer to as the ovarian cycle. Oops. And the ovarian is one of the many cycles we will be talking about in females. So let's do that. Here is the scary, intimidating table that kind of puts all of the pieces of all the female cycles together. What we're going to be focusing on first is right here. What is going on with the follicle? We know this follicular development is also associated with oogenesis. We've talked about how what's going on with there. These processes, as we've already hinted at and we'll see in much more detail, are controlled by the hormones of the anterior pituitary, primarily follicle-stimulating hormone and luteinizing hormone. So we will talk about what's going on with those levels. They don't only just influence the ovary to grow and release, but remember it also stimulates the follicular cells and the corpus luteum that is produced after that to produce our hormones. Two classes of hormones, estrogens and progestins. Remember, progesterone is just the most common progestin. I don't know why they always do it that way, but they do. And these, oops, wrong button. These estrogens and progestins are going to influence many things that are going on inside of the female, but the one most closely related to their cycle and most obvious of those related to their cycle is the influence that it has on the uterine wall and what's going on in the uterus. So what we have to do is, now that we've talked about oogenesis and what's going on there, we need to start talking about what's happening in the follicles, then we'll add the hormones, then we'll add the uterine cycle, and put all those pieces together, all right? Lexus versus bug, as we talked about. The female is far more complicated and more interesting than the males. Now, what I will say is that I think your book, and again, I'm constantly picking on artists, constantly picking on illustrators, constantly picking on the models. I will say I think your book has done an excellent job of describing this process. However, the one thing I will say, and again, it never occurred to me, but I've had numerous students who were confused by this, so I want to make sure to emphasize this here. And again, for most of you, it'll be a duh thing, but for some of you, it'll be an aha thing. So again, it's worth taking the time. Your book does a great job of showing all of the 8 stages of the ovarian cycle. They're going to talk about it. Does a really good, nice job of describing the process. We're going to talk about all of that. And what they've done here on this illustration of the ovary is to show us every single one of these steps along the way. The two important things to remember about this is you are not going to see all of these stages in the ovary at the same time. All right, so it goes first step 1 and 2 and 3 and 4 and 5 and 6 that way. But the other important thing is that these structures, the follicles, do not move within the ovary. So it's not like a clock where they start at noon and then move to 3 and then are ovulated at 6 p.m. and then rotate around. So what happens is this particular follicle right here may go through all of the stages of our ovarian cycle right here. And then next time, this one's going to do it. And next time, this one's going to do it. And it'll do it all at this location. And the next time, maybe it's over in the other ovary, right? And not in this one at all. So these clusters, these structures do not move within the ovary. They don't rotate from position to position. It is just showing the sequence of the events in here. So again, I've had enough people confused by that I want to mention that off of the bat. But otherwise, I think it's an excellent, excellent illustration that does a really nice job of describing all of the different processes. So let's start with the ovarian cycle. Our development of our ovarian cycle occurs in 2 phases. The follicular phase, which is D1 through D14, and the luteal phase, which is day 14 through day 28. Notice, and again, this is an important caveat to make right off the bat, we are using a quote-unquote average cycle length of 28 days. Now, as I look up at the list, I see that there are all of 13 people here today, and at least one of them, I'm guessing, is male. So we probably have at best a dozen females here. I haven't bothered looking at the participants to see just how many. I guess I can do that real quickly. We have a couple of boys in here. So again, while we use that typical number of 28 as the average cycle, is there anybody actually in the class who's willing to admit their cycle is precisely 28 days? Obviously, there's going to be some variation in this. Some can be as little as 21. Some can be as long as 35 or 40 days. So there can be some variation in it. In most cases, it doesn't vary from month to month. So it's not like a female is typically 25 days 1 week, and the next month she's 40 days, and the next cycle is 15 days or something along those lines. But there is typically individual variation in this. What's most important is the dividing point. What's the big major event that occurs that distinguishes the follicular phase from the luteal phase?
Ovulation.
There you go. Ovulation is the big key event. And I will tell you right now that of the two phases, the follicular is the one that is most variable. So fluctuations that women see in their cycles are typically, some have a shorter follicular phase, some have a longer follicular phase. But once ovulation occurs, typically the luteal phase is about 2 weeks. That part is fairly consistent for most females. All right. As we saw from the pretty picture that on the previous page, this ovarian cycle can be divided up into 8 steps. Again, do you have to precisely describe 8 steps as you're going through this? No, again, as long as all the information is there, that's what I care about. But in this case, 8 steps is a pretty good description of the different stages along the way. And what this starts with is our primordial follicle. If you remember back to, an hour ago, as we were talking about how we have that oocyte, and we have its follicle that surrounds and supports it. And if you remember, we said there are 3 types of cells. In the follicle. And those 3 types, actually I'll sneak this information over here. Those 3 types are follicular cells, granulosa cells, and thecal cells. So now we get to define what those are. We talked about this a little bit when we got to our endocrine system, but let's talk about it in more depth now. And let's see what's the best way to do this. Let's go with this. Excellent. When a follicle begins, when it is first produced in the ovary, basically that oocyte has a single layer of squamous cells surrounding it. So we have a single layer of squamous cells. That surround it. And that single layer of squamous cells are called follicular cells. So those are follicle cells. Then what happens is that egg starts to mature. Well, we can do that. That'll work. As the follicle starts to mature, that one layer of squamous cells becomes one layer of cuboidal cells. And then that one layer of cuboidal cells becomes 2 layers of cuboidal cells. And that 2 becomes 10, and so on and so forth. These cuboidal cells are a cube shape. So that's obviously one of the big differences. But the second difference with these cells, these cuboidal cells, is that the cuboidal cells produce estrogens. And so what happens is we will get 1 or more layers of these cells that will form and produce our estrogens. And these cells that form and produce the estrogens are the mature follicular cells, and we call them the granulosa cells. And that brings us to our thecal cells. Thecal cells are a different type of cell. Notice the granulosa cells are just mature follicular cells. They're follicular cells when they're immature. They are granulosa cells when they are mature. But out of the fibrous connective tissue, out of the tunica albuginea, an additional layer of cells that are different from the other will form on the outer layer. And these are our thecal cells. And these thecal cells that form, if you remember when we were in our endocrine system, they produce androgens. Those androgens play a limited role in sex drive, a limited role in aggression, but primarily they are used by the granulosa cells. Because while we say the granulosa cells produce estrogens, what they really do is these granulosa cells convert androgens into estrogens. So that's really what our granulosa cells do. So if you want estrogens, you have to have those androgens. So the thecal cells produce the androgens, they give them to the granulosa cells, and the granulosa cells then produce the estrogens. All right, so these are our three different types of cells, and we're going to go over this again as we look at them, because the number of cells, the type of cells determine what kind of follicle we have. And that's where I'm going to cheat now and get rid of all of this. Oh, this is going to be brutal to have to delete all of this. Oh, well, I'm already committed. Back to our original structure, and in this case, let's be specific, primary oocyte that is frozen in prophase 1. That primary oocyte frozen in prophase 1, surrounded by a single layer of squamous follicle cells, is what we call a primordial follicle. So if you think about it, from birth to puberty, the ovary is filled with nothing but primordial follicles. And they aren't just randomly distributed. They're typically clustered together. Let's go ahead and throw that word typically on there. And what do you call it when you have a cluster of eggs? They're clustered together in a nest. So we have these nests that form clusters of our primordial follicles located inside of our ovary. And that's our starting point. So we have nothing but primordial follicles as far as the eye can see, right? The ovary is filthy with them. And that's our starting point. Here we see an example there and here, and here's a partial one, here's this one. These are nice because you can actually see the chromatin inside that primary oocytes frozen in prophase 1. And again, if I were to point an arrow at this during the exam and say, identify this cell, you will hear me, or not hear me, but you will see those two magical words, be specific. And when I say be specific, that of course means to be very precise. I want all of that. That cell right there is a primary oocyte frozen in prophase 1. That is how you would identify it. And you can see some of them clustered together in this nest. These are our primordial follicles. However, notice one of these things is not like the other. Here we have a cell, and guess what? This cell is a primary oocyte frozen in prophase 1. But if you notice, something is different about its follicle. Those squamous cells have started to mature and are no longer squamous, but now cuboidal. And when we have a single layer of cuboidal cells, we no longer have a primordial follicle, but we now have what we call a primary follicle. And so that's our goal. During the ovarian cycle, we want to take a handful of these primordial follicles and start the maturation process. Remember, only one's going to be released. But during the ovarian cycle, we begin the maturation of these primordial follicles. And not just one, but numerous. All right. Here we see another great example of a primary follicle. We can identify it because it has a single layer of cube-shaped cells around our cell. And this cell, again, is still a primary oocyte frozen in prophase 1. It can take one of these primordial follicles over a year to reach this primary stage. So again, we think of the female cycle as being 28 days, but really, as I've said, it's a lot more complicated than that. The process of developing primordial follicles into primary follicles may take a very long time. But at the beginning of every cycle, a few dozen reach the primary follicle stage. Again, a primary follicle is this entire structure. The primary oocyte frozen in prophase 1 and the single layer of granulosa cells that are around it. Yep, I got a secret for you too. It also primary oocyte frozen in prophase 1 also applies to secondary follicles as well. So we're getting there. Notice the cell's much bigger, but it is still a primary oocyte frozen in prophase 1. Our cell has gotten bigger, but it still has not completed meiosis 1 yet. So again, this process of forming primary follicles is a slow process. A few dozen reach this stage. And notice, as we said, there's two big differences about the follicle. One is that they're now cuboidal cells, so they're those granulosa cells. But remember those granulosa cells can start to produce estrogens. So as we are getting the formation of granulosa cells, we are getting the production of estrogen. So estrogen levels are starting to go up. I want you to notice one other thing that is starting to develop here, actually two other things that are starting to develop here. The first is the candy-coated shell. As I mentioned, our egg needs to be protected. To protect this egg, we are going to put a glycogen-rich layer on its outer surface, a literal candy-coated shell. Of course, as fun as candy-coated shell is to say, we have to use the appropriate name for this glycogen-rich hardened layer, and it is the zona pellucidum. So our zona pellucidum is starting to form, protecting the egg. Notice also, and I'm going to go ahead and erase my circle so we can see this a little more clearly. Remember, these granulosa cells were the follicular cells. But notice these cells from the connective tissue are starting to condense on the outer surface of this. And these cells that are condensing on the outer surface are the thecal cells that are forming. And remember, these thecal cells are necessary because they produce the androgens And once it produces the androgens, then those granulosa cells can use those androgens to make estrogens. Which, convenient enough, it says right there. Now, as these cells start to produce estrogens, remember, as we talked about, estrogens cause the maturation of the oocyte and the maturation of our follicle. So what starts as a single cell with one layer of cuboidal cells on the outer surface becomes a small handful. Remember, we started with over a dozen. We get a small handful of primary oocytes, frozen in prophase 1, that have 2 or more layers of granulosa cells. And these primary oocytes, frozen in prophase 1, that have 2 or more layers of granulosa cells around them become our secondary follicles. And at this point, this is important. This point, the race is on. A small handful reach this phase. However, only one is going to win. Only one will be ovulated. Any other primary oocyte that gets to this secondary follicle stage, if it doesn't get ovulated, it gets destroyed. It basically dies and gets reabsorbed by the ovary. So at this point, you either win or you die. You are either ovulated or you're broken down and reabsorbed. This is your one shot, your one chance. So a dozen or so, a handful of these start this process. Start forming these, getting bigger. That secondary layer form, and it grows bigger and bigger and bigger. But only one of it makes it to the stage of maturation. All of the rest are going to degenerate. The reason for this is really interesting. Do I have it here? I don't. Perfect. So I can write it out. Excellent. We're going to talk about the hormones in great detail a little bit later, but one of the important things to understand is that estrogen has an interesting relationship with our anterior pituitary. At low levels, estrogen inhibits the release of follicle-stimulating hormone and luteinizing hormone. Notice there, just release, not the production. The anterior pituitary will continue to produce it, it just won't release it. All right? However, at high levels, estrogen triggers the production and release of luteinizing hormone. Well, let's go of follicle-stimulating hormone and luteinizing hormone. So at high levels, it's actually excitatory. And that's what happens with this race. So let's think about what happens. We have several secondary follicles forming. And as those several secondary follicles are forming, we get an increase in estrogen levels. Now again, remember, let's go back up to the top. These follicles are developing because of follicle-stimulating hormone. As the name indicates, follicle-stimulating hormone stimulates the follicles to grow. So we have several follicles growing, getting to this stage, and they're producing estrogen, and estrogen levels increase. The problem is this estrogen inhibits the release of follicle-stimulating hormone. So now there's less follicle-stimulating hormone available to stimulate the follicles. And without enough follicle-stimulating hormone, the follicle dies. So only those follicles that one of two things happens, right? Obviously, when follicle-stimulating hormone is released into the ovary, not all parts are going to get a completely even amount. Some randomly will get more than others. Those areas that get more, their follicles are more likely to survive. But also, as it turns out, each follicle is different in its sensitivity to follicle-stimulating hormone. Some are super dependent on follicle-stimulating hormone. So if follicle-stimulating hormone levels drop even a little bit, that one's toast. Others are less sensitive to changes in follicle-stimulating hormone. So as long as there's a little follicle-stimulating hormone left around, they're able to thrive, they're able to mature. So because of differences in amounts and differences in sensitivity, only one wins. And winning in this case means reaching the mature follicle stage. So like I said, once we reach this secondary follicle stage, the race is on. You are either going to mature, and if you mature, you get to ovulate, or you're going to be broken down and reabsorbed by the ovary. Now, notice this is steps 3 and 4 in your textbook. And the reason for this is there is one other change that starts to take place in our follicle during this phase. Notice we defined a secondary follicle as a primary oocyte. And again, I know that's confusing. This is a secondary follicle, but this cell is a primary oocyte frozen in prophase 1. That has 2 or more layers of cuboidal cells. So first 2, and then 3, and then 5, and then 10. Our oocyte is growing, but our follicle grows more. And that's actually what we see here. Notice what happens is, and here we can see these layers. Here's our thecal layer much more clearly. Our zona pellucidum is very distinct. This is still a primary oocyte frozen in prophase 1. But notice the follicle is starting to grow so big that gaps start to open up inside of it. I think I have one more picture. Nope. Small spaces, small fluid-filled spaces will start to form. These fluid-filled spaces are what are known as the antrum. This antrum will start to form as our follicle expands, because our follicle expands faster and bigger than the oocyte does. And so what will happen is these little pockets of fluid-filled space will start to open up. Notice at first they're about the size of the oocyte or smaller. But eventually these small antrums fuse together. And when these small antrums fuse together, they form one big large antrum. And when we have one big large antrum, right, where the antrum is now bigger than the size of the egg, then we now have our tertiary follicle. So again, notice they're calling this a late secondary follicle, whereas the previous one is just an early secondary follicle. That late just indicates that these antrums are starting to form. So again, over 20 primary follicles start to form. Only a small handful get to the secondary stage, And from here, only one. All right, there can be only one. Only one follicle reaches the tertiary phase. Tertiary follicles are also known, and I think I have the pretty words for them here, mature follicles. They're also known as Graafian follicles. All of those are acceptable terms. And there are two key things to know about this tertiary follicle. As we said, it has a massive antrum. That's how we distinguish it from the secondary. Secondaries may have small pockets of it that form, but this is going to have a massive antrum. But here's the other big thing that has occurred. By the time we reach this tertiary follicle stage, finally, our oocyte has completed meiosis 1. And of course, if it's completed meiosis 1, now the cell we see is a secondary oocyte frozen in metaphase 2. Again, tertiary follicle, but so this whole thing is a tertiary follicle, but this cell is a secondary oocyte frozen in metaphase 2. Exactly. All right. So our follicle continues to develop. Again, clearly see the thecal layer. Clearly see that zona pellucidum that we talked about, that acellular layer. Notice, though, one other thing. Because of this big, huge antrum, and I'm going to cheat and use my highlighter. Blue works. Notice we have kind of a division of our granulosa cells. All of these cells right here are granulosa cells. All of these cells right here are granulosa cells. Is there any difference between this cell right here and this cell right here? Any difference in its function, any difference in its anatomy, any difference in these two cells at all?
The chromatomes or chromosomes in the middle?
Nope. They are identical cells. The only thing that is different is their location. All right, that's the only thing that's different. These cells are still in contact and surrounding the egg. Remember, the oocytes' chromosomes are different, but these granulosa cells are not. These granulosa cells are granulosa cells. They're producing estrogens. But this particular granulosa cells surround the egg, kind of like a crown around its head. And that's where they get their name. These granulosa cells that are surrounding the egg, in direct contact with the egg, are known as the corona radiata. Because as we all now are intimately aware, corona means crown. The rest of these are also granulosa cells, but they're just the follicle, right? Because they're not in direct contact with the egg. Here in the tertiary follicle, they're serving the exact same function. However, when ovulation occurs, it is only the egg and the cells that are forming the corona radiata that will be released. Only this structure is expulsed. And as we talked about, the remaining follicular cells that stay behind are what become the corpus luteum. So, about 14 days into this process, into this cycle, one sole survivor has made it to the mature follicle stage with this massive fluid-filled antrum and that corona radiata that is formed around it. And like I said, at this point, our oocyte has completed meiosis 1, started meiosis 2, and frozen in metaphase 2. All right, frozen in metaphase 2. At this point, we have a massive follicle. And at this point, we're producing massive amounts of estrogen. And remember, as we talked about, estrogen has that complicated relationship with the anterior pituitary. Once estrogen reaches a critical point, it actually triggers the release of our follicle-stimulating hormone. But more importantly, in this case, it triggers the release of that massive amount of luteinizing hormone that has been accumulating inside of the anterior pituitary. And that massive spike in luteinizing hormone triggers ovulation. Here we actually see that mature follicle actually bulging out the side of the ovary like a big balloon. And like a big balloon, it pops. And when it pops, that egg with its corona radiata is expulsed. There's a couple implications to this. The first is when you pop that balloon, it does leave a little bit of scar tissue. If you just happen to be walking down the street and randomly find an ovary, if you look at the surface of the ovary, you'll see scarring, you'll see pitting, one for each ovulation. And by counting the number of scars on the outer surface, you can get an estimate of how many times that ovary has ovulated, and you can get a good guess at how old the individual was who lost it. And that makes it easier to return the ovary to its respectful owner. The other interesting thing about this is this rupturing is a tearing of the tissue. And there are some women who are actually sensitive to the sensation of it. It's got a great a German name called middle spurts. That sensation, what happens is some females, I don't know the actual percentage, but some females may feel a kind of minor pinching sensation inside. And as it turns out, that happens to be the sensation of ovulation occurring. For many women, it occurs and they're not even aware of it. One of my favorite stories of this is when I was first started teaching, I was an adjunct teaching summer school over at Woodland Community College, and the department chair there was trying to get pregnant. Her and her husband were trying to get pregnant, and of course, when you're trying to get pregnant, one of the things you do is you monitor your cycle so that you can try to copulate at the correct time to get that. At the time, she was 35, 36, so a little old in the process, which is why they were getting all this help. And it wasn't until she was actively monitoring her cycle that she realized that that minor pain she felt, every once in a while, when she noticed it, was ovulation. She actually had the sensation she could feel ovulation when it occurred. She had just never been aware of what it was because she had never paid it much mind and never thought about it. It only happens about once a month, right? And maybe you were turning or twisting, or who knows what happens when it happens, and so you don't think twice about it. Right? Of course, once you realize that you can feel when you ovulate, getting pregnant gets a lot easier. And not surprisingly, she was successful getting pregnant and has had actually 3 children since then. So again, you have that ability. Some women have that ability to be able to perceive it. Not all of them, but some women are able to do that. Again, this rupturing of the follicle is triggered by that massive release, a massive spike, of luteinizing hormone that was triggered by estrogen levels reaching that critical point. When that occurs, our secondary oocyte, frozen in metaphase 2, remember it's still immature at that point, is expulsed into the abdominal pelvic cavity with its zona pellucidum, with its corona radiata. But again, remember, the majority of the follicle cells, and again, granulosa cells, I guess, would be more important. Granulosa cells that form the follicle remain behind. And again, the reason that's important is the second function of that luteinizing hormone. Luteinizing hormone luteinizes the remaining follicle, and it converts those cells, that structure, into the corpus luteum. Now, once that oocyte is expulsed into the abdominal pelvic cavity, like we said, things like the fimbria convert currents in it in hopes of getting it into the uterine tube. But remember, it does not have to enter the uterine tube. It doesn't always enter the uterine tube. That is the goal, and there are things that we do to try to ensure that that happens. But it isn't always necessary. It's not a certainty. At this point, and I guess I'll go ahead and clear all of that, our follicular phase ends, and thanks to luteinizing hormone, we begin our luteal phase. Our luteal phase, and again, here we see that with the pretty picture here from our textbook as well. The luteal phase is when those remaining cells of the follicle are converted by that luteinizing hormone into a big glandular structure called the corpus luteum. It's one of our endocrine glands, if you remember. And the big difference between the corpus luteum and the follicle is that while the follicle just produces estrogens, our corpus luteum produces estrogens and progestins. Progesterone, our primary progestin, remember, is that warning bell hormone, basically telling the female body, hey, the egg is on the loose. Pregnancy could occur any minute now. Get ready. And so it's that warning bell hormone that tells the female's body ovulation has occurred, fertilization may occur, and we need to get ready. Remember, that egg is only viable for 24 hours after it is expulsed from the ovary. So it needs to be fertilized within 24 hours after expulsion. I don't remember if we said it or not, but sperm are viable for about 48 hours, although there are some, and again, this is not my term, this is the appropriate term for them, what are known as super sperm, and those super sperm can be viable for 72 hours. So basically, you have about a 3-day window from when copulation can occur to have successful fertilization. However, while that egg needs to be fertilized within the first 24 hours of it being released, it can take up to 10 days before it implants in the uterine wall. It's going to take a leisurely journey down the uterine tube to the uterus and even hang out in the uterus for a few days before it implants. So that's what we need the progesterone for. Those progestins tell the female body, all right, anytime within the next 2 weeks, you're going to have an egg that's going to be in a fertilized egg in the uterus that's going to want to implant to become a baby. So we need to get ready. And so that progesterone is that huge hormone, that warning bell that tells the female's body to get ready. Of course, if 2 weeks go by and fertilization didn't occur, well, then all that warning bell was for naught, right? In which case we expulse the uterine wall and we start the whole process all over again. And that's what happens. We produce this big, huge, massive corpus luteum, but it turns out the corpus luteum is sensitive to luteinizing hormone. And as we know, estrogen levels are going to drop. Estrogen inhibits our luteinizing hormone. So our luteinizing hormone goes away. And as our so let's write that up here. Levels of luteinizing hormone they first spike, but then decrease. And as they decrease, our corpus luteum decreases as well. And therefore the amount of estrogens and progestin produces decrease as well. After about close to 2 weeks, what happens is our corpus luteum basically goes from this big, huge, dark glandular structure to a shriveled up piece of scar tissue. And this shriveled up piece of scar tissue is called the corpus albicans. And the corpus albicans is no longer glandular. So no corpus albicans, our levels of progestin and estrogen decrease rapidly. Right, that's going to lead to menstruation and other things, but once estrogen levels drop, our anterior pituitary can start producing follicle-stimulating hormone and releasing follicle-stimulating hormone again, stimulating our primordial follicles, and the whole process begins again. Unless fertilization occurs. If fertilization occurs, that messes everything up. But we'll get to that later. But assuming fertilization does not occur, the drop in estrogen and progestins allow the anterior pituitary to start producing follicle-stimulating hormones again. That stimulates the primordial follicles to start to become primary follicles. And then again, those primary become secondary, the race is on. And again, that process continues. Around and around she goes. So again, here we have that pretty picture that sums it all up. Again, the follicular phase, we go from primordial to primary to secondary. Right, here's another secondary. Notice those antroms are starting to form, but it is until they fuse together to become one big, huge, massive antrum that we have tertiary. Remember also at this tertiary, we finally have completion of meiosis one. So primordial, primary, primordial, primary, secondary. All of these are primary oocytes frozen in prophase one. But here in our mature follicle and during ovulation, we have secondary oocytes frozen in metaphase 2. The remaining cells of the follicle are converted into the corpus luteum by our luteinizing hormone, which then goes away and it degenerates into the corpus albicans, and then the process begins anew. So again, follicular phase, primordial, primary, secondary, tertiary. Then ovulation occurs, and we have our luteal phase. Formation of the corpus luteins, corpus luteum, and then degeneration of the corpus luteum to ultimately form the corpus albicans. And that, just that simply, is our ovarian cycle. Here we see the pretty picture associated with all of that. And notice, again, we'll talk more about hormones in a bit, but again, if we cheat, we can see it as that spike in luteinizing hormone that triggers ovulation, the change from blue to green, which is our follicular phase versus our luteal phase. So we've focused here. We've hinted at what's going on with ovarian hormones. We've hinted at what's going on in the anterior pituitary, but we'll go over those again. So we've primarily focused on what's happening here in the follicle and related it to the process of oogenesis. All right, questions on that? All right, we still just dipped our toe in the water. There's still a fair amount of things that need to be talked about and discussed, but I think this is another good natural stopping point. So as I look at my watch, I see that it is 10:32. So let's go ahead and restart. At that would be 10 what I say was 32, so 10:42. And again, I will start recording at that part. All right, so any questions before we take our next break? Hopefully this makes some semblance of sense. If it doesn't, it's just going to get worse. So again, the females is layer upon layer upon layer. If you don't understand the foundation, then it's going to make it even harder as we try to add more layers to it. So if there's any questions, please, please ask them. All right, then we'll go ahead and pause, and I'll meet you back here in 10 minutes. And away we go. So we have talked about the ovarian cycle and what goes on in there. And obviously, as we talked about, one of the keys to this, as we saw, is that it is controlled by hormones and related to hormones from the anterior pituitary. However, again, this isn't a process that is beginning at birth. So obviously something has to transition, changes have to take place during puberty that cause the establishment of the ovarian cycle. And again, it comes back to this complicated relationship that estrogen has with the anterior pituitary. In that adolescent, in that infant child, female, her ovaries are secreting small amounts of estrogen and small amounts of inhibin, and those are inhibiting the hypothalamus and inhibiting the anterior pituitary so that it is not producing follicle-stimulating hormone or luteinizing hormone. So as there is little to no follicle-stimulating hormone and luteinizing hormone in the female's body early on. However, at the onset of puberty, leptin actually stimulates the hypothalamus. Anybody remember where leptin came from? What are the cells? What is the tissue that produces leptin?
Adipose.
Adipose, excellent. Adipose produces leptin. Again, if you think about this from a biological standpoint, this makes sense. If a female is going to produce offspring, again, it isn't like the male where all she has to provide is the chromosomes. She has to provide the chromosomes, but she then also has to be able to house and has to be able to maintain that growing baby inside of her body for months. To be able to do that, you have to be physically fit, physically strong, physically healthy enough to be able to do that. And one of the primary ways you can tell your body, hey, I have the resources necessary for the massive increase in metabolism necessary to be able to produce offspring, by having enough nourishment where you produce adipose tissue. So having adipose tissue tells the body, I am healthy enough to be able to house and metabolize an offspring. And so that adipose produces leptin. You see the flip side of this as well. And again, we are talking about reproduction when we talk about healthy, when we talk about all of these processes. Obviously, there are extreme athletes, some of these extreme runners who just for hoots will see just how far they can run in a 48-hour period of time, right? They spend an entire weekend spent running hundreds of miles. Obviously, they are amazingly physically fit and active. However, what can sometimes happen with some of these extreme athletes, the triathletes or the extreme marathon runners, is that their adipose levels get so low, they get down to 0.5% body fat in some of these extreme types of conditions like that. And what actually can happen for some of these extreme athletes is it can actually disrupt their cycle. That decrease in adipose decreases the leptin. It can actually disrupt their normal, at that point, mature cycle. So the onset of adipose, and again, it's speculation at this point, but it's educated speculation. One of the current trends that we've seen, especially in industrialized nations, is a onset of puberty earlier in adolescence. So again, it used to be closer to 13 or 14 when it would start. And now, like I said, it's down to 12, and in some cases even 11 or 10 years old, that the first menses can take place. And some speculation of that is, again, the overall health of us as a society. We tend to be better nourished now than we were 100 years ago. And so again, being able to have more adipose stimulates this process sooner. The other thing, too, if you think about it, again, it is, it's not impossible, but most milk you buy these days is milk that is not treated with hormones. Of course, they've got that big important message on there. You know, studies have not shown that RSBTS or whatever it is doesn't have any effect on the body, but most milk companies are now not using that hormone to produce milk production because there was some speculation that those growth hormones were also what was leading to an early onset of menses in females as well. Those same feminizing hormones they were using to increase milk production were affecting the people drinking the milk. So onset of leptin, having the body healthy enough, stimulates the hypothalamus. And now the hypothalamus can start producing its gonadotropin-releasing hormones. And from that, we start to, of course, and let's sneak it in here, we start to see an increase in production of follicle-stimulating hormone and luteinizing hormone. These hormones, the gonadotropins, then slowly start to increase. Typically at first, these cycles are highly irregular. A female may have her first menses and then may go months before she has her next one. And they can be longer or more variable in the timing of them. But typically, eventually, the adult cycle is established, and that could take anywhere from 1 to 3 years for their eventual adult stabilized cycle to form. Now, one of the things that, again, I want to emphasize is that during this irregular cycling, it is not uncommon for a female to not ovulate an egg during that time. Wait, I'm not sure how that, I may have had too many negatives in there. So let's say that again. It is common during this establishment of the cycle that occasionally the female will not ovulate an egg during the cycle. So she is not guaranteed to ovulate a cycle during every menses or after every menses during the establishment of the cycle. But unfortunately, there are plenty of instances of that 12-year-old getting pregnant because ovulation can occur. It's not guaranteed to occur, but it can occur during those times. All righty. So now that we've established the adult cycle, let's talk about that adult cycle. And again, a lot of this is stuff we hinted at when we were talking about the ovarian cycle, but I want to make sure we emphasize it here. Our hypothalamus produces its gonadotropin-releasing hormone, and that stimulates the anterior pituitary to produce and release follicle-stimulating hormone. And so what happens is you see a gradual increase in the amount of follicle-stimulating hormone. And again, both are being produced, but remember, during the follicular phase, and let's sneak this in here, it is the follicle-stimulating hormone levels that are more significant and more impactful. That follicle-stimulating hormone stimulates the follicles to grow. And we go from primordial to primary to secondary. The problem is, as those follicles grow, the amount of estrogen being produced and also the amount of inhibin being produced by the ovary increases as well. And as we talked about, as estrogen levels increase, at low levels, they inhibit the release, not the production. Follicle-stimulating hormone and luteinizing hormone are still produced. By the anterior pituitary, but they are not released. And so what happens is the levels, that didn't work, follicle-stimulating hormone stabilize and even can drop. And as we talked about, that drop in the level of follicle-stimulating hormone is what leads to the attrition of our secondary follicles. So those that either don't get enough or those that are too sensitive to changes in the levels are eliminated, so that ultimately only one follicle reaches the mature state. However, that mature follicle is massive. And that mature follicle is now producing very high levels of estrogen. And when it reaches that critical point, then that estrogen actually triggers the massive release, right? Rapid and massive release of follicle-stimulating hormone and luteinizing hormone. But again, this time, It is the luteinizing hormone that is having the more significant effect. And that luteinizing hormone, that massive, huge spike of luteinizing hormone, triggers ovulation. But remember, that massive spike of luteinizing hormone really has two effects. The first is that it triggers ovulation. But the second effect is that that luteinizing hormone transforms the remaining granulosa cells, that ruptured follicle. It converts those into the massive glandular structure that is the corpus luteum. And the big difference between the corpus luteum and our follicle cells is that the follicle cells just produce estrogens, whereas our corpus luteum produces estrogens and androgens. Pardon me, estrogens and progesterones, progestins. Why does that say progesterone? It should say progestins. I shouldn't make that mistake, but I did. Oh well. Of course, progestins and estrogen inhibit, in this case, both production and release of follicle-stimulating hormone and luteinizing hormone. So this time, not only are we not releasing it, but we're storing it inside. This time, we're not making it and not releasing it. So levels of follicle-stimulating hormone and luteinizing hormone decrease. And remember, it's that luteinizing hormone that doesn't just produce the corpus luteum, but it maintains the corpus luteum as well. So if our luteinizing hormone goes away, our corpus luteum goes away. And as our corpus luteum goes away, our estrogens and progestins go away. Until ultimately, our corpus luteum goes away entirely, shrivels up, becomes the corpus albicans. And when it becomes the corpus albicans, estrogen and progestin levels are so low that our anterior pituitary is not blocked anymore. And when it's not blocked anymore, now our anterior pituitary can start producing follicle-stimulating hormone and luteinizing hormone again, and they start stimulating the cycle, and the process continues anew. Again, we have this pretty picture. Well, we actually have two pretty pictures. Here's a pretty picture that kind of shows this process of how it is going on in the ovary. But really, I want to come back to this one here. Again, here we have our ovarian cycle. And here we have our hormone cycles of the hormone levels in the anterior pituitary. Again, notice at first, follicle-stimulating hormone and luteinizing hormone levels are increasing. So we get that increase that occurs. But as that increase occurs, we start getting the maturation of the follicles. And as we get the maturation of follicles, remember this is going to cause an increase in estrogen. That increase in estrogen inhibits the release of follicle-stimulating hormone and luteinizing hormone. So remember, notice as we talked about, those levels, those level off and can actually go down. And remember, it is this decrease in follicle-stimulating hormone that leads to the attrition of the follicles. Until one and only one remains. That one remaining follicle is our mature follicle. And remember, this mature follicle is producing massive amounts of estrogen. And when it reaches that critical point, then that estrogen now triggers the massive rapid release. Notice again, both follicle stimulating hormone and luteinizing hormone have a massive release, a massive rapid release. But remember, in this case, it is the luteinizing hormone, which A, has the bigger spike, but B, more importantly, it is what triggers ovulation. But the other thing this luteinizing hormone does is it triggers the transformation of these follicular cells, or really granulosa cells, the cells of the follicle, the granulosa cells, are converted into the corpus luteum. And this corpus luteum produces estrogen and progesterone. I'm just going to put E and P here because I'm running out of space. And estrogen and progestin together inhibit both production and release of our luteinizing hormone and follicle stimulating hormone. But as luteinizing hormone levels go away, our corpus luteum goes away until it finally shrivels up into that non-glandular corpus albicans, at which point we are no longer producing high levels, well, let's just say estrogen and progestin, at which point the inhibition goes away. And when that inhibition goes away, we start getting an increase in production, and that one should actually probably be a different color. We get an increase in production of follicle stimulating hormone, and the process begins all over again. So we talked about it when we did the ovarian cycle. We talked about it when we talked about the ovarian cycle's hormonal control. And here we have the pretty picture that shows the process as well. And again, if we clear this image, we can then put it together with this one, which shows the hormone levels of what's being produced by the ovaries. Again, at first, estrogen levels are very low. That's why follicle stimulating hormone is able to be produced. But as we start to produce primordial to primary, primary to secondary, there is a mild increase in estrogen. And this increase in estrogen, again, inhibits release, but not production, until we get that nice big large primary follicle that is producing massive amounts of estrogen. And once it reaches that critical point, that triggers that spike of luteinizing hormone, which triggers ovulation. Notice when ovulation occurs, our follicle is disrupted, right? You're destroying that or damaging the cells. Not surprisingly, hormone levels drop. But that luteinizing hormone is also converting those cells of the follicle, those granulosa cells, into our big glandular structure, the corpus luteum, that now, for the first time, is producing progesterone, right, or progestins. Notice it produces estrogens as well, but not as high as a level. So these progestins are the ones that are the warning signal to the body, hey, uterus, get ready. Fertilization can occur any minute, but as the luteinizing hormone goes away, the corpus luteum goes away. As the corpus luteum goes away, progestin and estrogen levels drop. And once they drop, the inhibition ends and the whole process begins anew. So notice we have added all of the pieces to the puzzle. What we're left with, the last little bit of the cycle we need to talk about, is what is happening in our uterus. All right. Questions on that? So again, this was scary and intimidating when we first looked at it, but now we've mastered 3/4 of it and it ain't so scary no more. Excellent. All right. Now, we have talked about what's going on in the ovaries. We haven't talked too much yet about what happened to that secondary oocyte frozen in metaphase 2. And let's go ahead and write that just to beat that dead horse. That secondary oocyte is frozen in metaphase 2 is the state that it is in when it is released via ovulation. The goal is to get it into the uterine tube, or what is also known as the fallopian tube. So again, that is our goal, to get it into there. And again, there are some specializations to try to help with it, but that isn't a guarantee. Remember also, this egg is not motile. It doesn't have swimming capabilities the same way that the sperm does. So it instead is going to require, to get into it, currents within the peritoneal fluids. And then once in, it's going to take muscle contractions and other help to move the egg along the way. It is a slow, arduous journey. It can take the egg as long as a week, usually about 5 days, but it could take as long as a week to get to the uterus. So let's say 5 is a good round number. As we said, that egg is only viable for 24 hours. 25 hours does not equal 5 days. So if it is going to be fertilized, that fertilization has to occur in the uterine tube, and typically occurs in an enlarged region that we see here, and as you can see here, is labeled as the ampulla. So this enlarged region here, the ampulla, is typically the site for fertilization. Then once fertilized, it can travel the rest of the way to the uterus and then ultimately implant. If not fertilized, then it degenerates and breaks down. If it doesn't get into the uterine tube, it degenerates and breaks down. Now, the uterine tube, as we mentioned, is part of the ductwork system for females. As we can see here, it connects to the uterus via this narrow tubed region at the end, known as the isthmus. Let's clear that. Isthmus through here. However, as we also mentioned, this is an open duct system. So it does not actually directly connect to the ovary, which now that we understand more about the ovary makes sense. You wouldn't want to just attach the uterine tube to the very top, because there's no guarantee that the very top is where ovulation is going to occur. Ovulation can occur at any point of the ovary, and so we don't want this unless it was going to completely encircle it, which I guess would be one way to do it biologically, but for whatever reason it didn't. And again, the person who designed this didn't leave us her blueprints or footnotes, so we don't know why she did what she did, but it doesn't connect directly to it. There are some specializations where you're not doing that. There you go. As we mentioned, there is the enlarged portion known as the ampulla, where fertilization will take place. From that is a funnel-shaped region known as the infundibulum. And coming off of the infundibulum are these extended finger-like projections known as the fimbria. These fimbria move in a wave-like fashion. And again, their goal is to produce currents in the peritoneal fluid, so that when the egg is ovulated, those currents are picked up and carried into the funnel-shaped infundibulum, which funnels it into the ampulla, where fertilization can take place. Again, this helps to encourage the egg to enter into the uterine tube, but it isn't guaranteed. Notice one other thing. The epithelial tissue of our uterine tube is ciliated. And when you see that term ciliated, you know, of course, that means this must be lined with a ciliated pseudostratified columnar epithelial tissue. And the answer to that is nope. Remember, in the epididymis of the male, we saw a unique tissue we had not seen before. There we saw a ciliated pseudostratified columnar epithelial tissue that was non-motile, cilia that did not move. Well, here in our uterine tube, we have a unique tissue as well. We have a ciliated simple columnar epithelial tissue. Normally, when we think of a simple columnar, we think of microvilli. They have microvilli that increases their surface area, and they are non-motile. Well, here in our uterine tube, we actually have simple columnar cells that have cilia. So that when that egg gets into the uterine tube, those cilia, along with peristaltic contractions, will move the egg along the path to our uterus. Again, it's a slow process. It can take almost a week, 5 days to get there. But those cilia of those simple columnar cells, again, a unique tissue. And again, it makes it easy to tell and identify this tissue because it's the only place that you're going to find this ciliated simple columnar epithelial tissue. But it's going to help to move that non-motile egg. As we said, when expulsed, it's viable for at most 24 hours, usually 12 to 24 hours after ovulation. And like I said, those cilia will move the oocyte towards the uterus. And like I said, it can take around 5 days, but can take as long as 7. So notice it says 3 to 4, but it could take 5 to 7 days even before it finally reaches the uterus. Clear those. When we talk about the anatomy of the uterus in a female that has never been pregnant before, it is about the size and the shape of a pear. So notice, and let's talk about the anatomy first. Do I have the anatomy? Yeah, let's talk about the anatomy first. About the size and shape of the pear. It has a head that sticks out, and that head that sticks out is known as the cervix. Notice the head of the cervix actually sticks out into the vaginal canal. In that head of the cervix, there is a narrow restrictive entryway known as the cervical canal. This is a restrictive space, often closed with a mucus plug, because again, this is an opening to the abdominal pelvic cavity of the female. So we want a restrictive opening closed off as much as possible. The bulk of the uterus is the body. As we mentioned, the two narrow ends are the isthmus, where it connects to the uterine tube. And the superior part of it is the fundus. Remember, the fundus of the stomach was something that sticks out. The fundus of the uterus is something that sticks out as well. One of the interesting things about the fundus of the uterus, especially in a female that has been pregnant, you can actually palpate the fundus of the uterus as the uterus grows. And actually, one of the things a doctor will do is with a tape measure, they will measure from the pubic symphysis up to where they feel the fundus of the uterus. And that distance is actually a fairly reliable indicator of how long the female has been pregnant. So by telling that distance from the pubic symphysis to the fundus of the uterus, you can get a fairly accurate estimate of how long the female has been pregnant for. All right, so those are the portions of it. When we look internally, and of course the function of the uterus is threefold. It receives that fertilized egg. It retains the fertilized egg, keeps it, embeds it in the wall of the uterus, and then obviously has to nourish and feed it and allow it to develop, containing it for the 40 weeks before it then expulses it out into the real world. When we look at the anatomy of the wall of the uterus, it is lined by 3 layers. The innermost layer that lines the uterine cavity is the endometrium. This is the part that allows for implantation. This is the part that grows elaborately, vascularizes elaborately, and glandularizes elaborately in anticipation of pregnancy. But this is also the layer that if pregnancy or fertilization doesn't occur, is then expulsed as the menses. We have a muscular wall, the myometrium. It is, of course, smooth muscle, producing the contractions necessary for three things: expulsion of the uterine wall for menses, expulsion of the baby during labor, delivery, or what we call parturition. And its other job, as we mentioned, is with the help of prostaglandins from the semen, it can produce a reverse peristalsis that can draw the sperm up and bring it to the egg for fertilization. Lastly, we have the outer layer, our serosa. The uterus is an intraperitoneal organ, so it is surrounded by a serosa, and of course that serosa would be the visceral peritoneum. Hold on one second. All right. With that, we have discussed the gross anatomy of the uterus. We haven't talked, we need to talk about some of the microscopic anatomy, but this is actually as far as I wanted to get for today. It is 11:15, so that's about a normal time. It's a little bit early, but in looking at the material we have left to cover, we are going to do an abbreviated version of the developmental stuff, so we're not going to go in quite as much depth as your notes do. You will only be responsible for what we cover in class, the little bits of it that we do cover. But this is where I wanted to call it a day, especially because we have a lecture Monday, 2 days before the exam on Wednesday, and then our final. I want to limit how much of the developmental stuff we need to talk about, because as I said, it's going to be a little bit more challenging. So I think this is a good stopping point. I think we've talked the gross anatomy, because once we talk the microscopic anatomy, we're going to want to talk about the uterine cycle, and that's going to take us far beyond what I wanted to do time-wise for today. So even though it's a teeny bit early, I don't think you guys will complain about stopping after just 3 hours and 15 minutes instead of going our 3:30 like we normally do, unless someone really, really wants me to continue. Feels they need to get their full 4 hours worth of me on this day. Then I think this is a good stopping point for now. Yes, so I did mention that at the beginning of class. Please remember, there will be a histology review. I'll actually put it on the schedule. We'll do it through the class Canvas, so Canvas Zoom. So instead of going to my office like we normally do, I'll actually schedule it on the Confer Zoom calendar. There is going to be a histology review on Friday from, 10 to 11? Yeah, from 10 to 11. So I will, as soon as I'm done here, I will schedule that so that you'll just access it the same way you access the lecture. Go to the Confer Zoom and put it there. And then that way it'll be on the calendar there as well. So yes, we will have a histology review on Friday. I've already actually made the slides for that, so I'm actually ready and prepared for us. All right. And again, some of those I actually stole from our endocrine because it was easy, because again, we talked about some of this stuff there as well. All right, perfect. That'll be our stopping point. I will go ahead and stop the recording.